Hi, everyone, and welcome back to the last keynote of the Bioconductor 2021 conference. I think many of you have already been through the technicalities, but just as a reminder, you can feel free to put questions in the Q&A box on the right side, and others can upvote those, and then we'll bring those up at the end of the talk. You can also put things, links, or other things in chat if you like, which are not questions. And also we can use the raised hands feature if you want to ask a question in person at the end of the talk, and we'll bring you on stage. So now I will introduce our speaker. Dr. Melissa Wilson is a computational evolutionary biologist. She has a PhD in integrative biosciences, bioinformatics, and genomics from Pennsylvania State University, supervised by Katarina Makova. And then she did a postdoc at Berkeley with Rasmus Nielsen. She's now an associate professor of genomics, evolution, and bioinformatics from the School of Life Sciences at ASU, and a co-director of the Computational Life Sciences program there. Dr. Wilson and her lab study the evolution of sex chromosomes. And with respect to human health, her lab studies why there are sex differences in health and disease, including in liver cancer and in human placenta, and how sex bias processes have affected patterns of genetic variation in human populations in Africa. Through her work in understanding features of sex chromosomes and phenotypic differences, she's outlined a set of improved practices for analyzing genetic data without discarding the important information with respect to sex chromosomes. And so I really appreciate myself having learned from, for example, like her Twitter threads about how to do this properly and not lose important information. This work and other aspects of inclusion of sex as a biological variable is in partnership also with the NIH Office of Research on Women's Health. And in this way, I'd say that Dr. Wilson has acted as a multiplier. So it's not just adding to the information, but not just improving the quality of work from her own lab, but removing blind spots in the field, and which otherwise might block us as geneticists or biologists from a complete understanding of mechanisms of human health. And I'll step down.
Thank you very much. That was a really wonderful and generous introduction. I recorded the talk because I have two small children at home, and I was not sure if they were going to be quiet. And so now I'm kind of deciding at the last minute that everyone's cooperating, so we'll start. But just as a heads up, it might just all go downhill halfway through this. And if so, I apologize. This is life. As a reminder, we are still solidly in a pandemic. But with that, I do encourage everyone, please put your questions in the chat or the Q&A, and I'm really happy to talk through all of this work. So yeah, let's get going. Share screen. Oh, sorry, just a minute. Sorry, I want to select PDF file. Nothing makes you feel old like new technology, but I guess that's the great thing about conferences like this, is you get to learn lots of new technology. Maybe a great thing for trainees is to, what is that? The more you learn, the more you realize you don't know. That's always true. And I feel like every day I learn a little more, and then I have more questions than I did before. So it's always good. Please do ask your questions here. So like Michael mentioned, you can find me on Twitter. I'm in the School of Life Sciences, the Biodesign Institute, and the Center for Evolution Medicine at ASU. And we'll just get going here. So one of the things I like to start with is that we want to be cautious with our language. So when we talk about sex differences broadly, we might be meaning a lot of different things. So me, trained as a geneticist, I often think about, in humans, individuals who typically have two X chromosomes, who typically pass on their DNA via eggs, and individuals who typically have an X and a Y chromosome and pass on their DNA via sperm. Some people may only be interested in the gametes that are being produced. Some people may be interested in gonads or gonadal development. Others might be talking about gender differences if we're thinking epidemiologically. And the thing is that these, while they overlap, they don't map one-to-one on each other and have independent effects on our health. And that's both fascinating and causes a lot of problems. And so today I'm going to be really cautious and talk to you really about the X and Y chromosomes, and try to be specific when I'm talking about XX or XY individuals, because when we're thinking about genome alignment, one's gender presentation or their gonads are less important for what I'm going to talk about today than their sex chromosome complement. And that's really important because not everyone knows their sex chromosome complement, or, as we'll get to, it may be different in some of the tissues in the body than most of the tissues in the body, right? Yay, genetics. Okay. Moreover, these sex differences are multimodal. So although I said typically we think about individuals with XX or XY sex chromosomes, 1 in 2,500 live assigned female at birth individuals only has a single X chromosome. 1 in about 500 live assigned male at birth individuals has 2 X chromosomes and a Y. So these are Turner and Klinefelter syndrome. Additionally, the genetics that are important for gonad development can have mutations across the genome on the autosomes, right? So we can have variation in our genetics, we can have variation in our gonadal hormones, we can have variation in gonad development, and additional dimensions, but one that's really important is time. So we know that over time, individuals, specifically XY individuals, will often lose their Y chromosome as they age in a proportion of their cells. Gonadal hormones shift dramatically from pre-pubescent through puberty, through menopause. So each one of these is multidimensional and varies across time. And I think that's really important. And we'll come back to the genetics, right? So someone may say, you know, I'm a woman, I'm a female, with genotype, I have XX chromosomes, but still in a tumor sample that they have, they may have lost one of their X chromosomes, or one of their X's was reactivated or duplicated, or any of these things. And so we want to try to be really cautious when we're doing the computational side of it to not make assumptions about the genetics of the sample relative to the identity of the person from which the sample came. Oh, and what I'm hoping to convince you, if you take one thing home, is that evolution matters for alignment. So this is a picture of the X chromosome and the Y chromosome, and these little blue tips are shaded. This is not exactly how large they are, but the blue tips are shaded here to show you the regions that are 100% identical between the X and the Y, and the green parts are the parts that have diverged between the X and the Y chromosomes. Beep. And so if I'm going to convince you evolution is important, I should tell you something about evolution. So we'll start with mammalian evolution. So these are mammals that were sequenced at the time that I got my PhD. And that was so long ago now that although there were about 32 mammals whose genomes had been sequenced at that time, now we have hundreds or thousands even of mammals with at least some information about their genome. And even these, I say their genomes were sequenced. They were sequenced to about 2x coverage, all aligned pairwise to human. And we have just come leaps and bounds from here. But I can't show you all of the mammals that have been sequenced to date. So we'll just start with these ones. And we'll walk through evolution of mammalian sex chromosomes, of which humans are, one arguably important mammal that we care about. So the first thing to think about is divergence of birds versus mammals, and that occurred about 300 million years ago. And then within mammals, there's 3 major groups of mammals that have dramatic differences in their sex chromosome complement. So the first group of mammals that is diverged relative to Therian mammals are the monotremes. So those are the platypus, and then there's about 5 species of echidna. These are egg-laying mammals, and they diverged about 177 million years ago. And then the marsupial and eutherian mammals diverged from each other about 160 or so million years ago. Marsupials are the pouched mammals, And eutherians are humans, dogs, cats, mice, whales, rats, dogs, pigs. A lot of the mammals that we study are both clinically or agriculturally relevant that we have there. So let's look at this phylogeny again, now that we understand the divergence, and look at their sex chromosomes. So birds have what's called a ZW system. If you've heard of XY or ZW, we use XY or ZW to refer to the sex that produces the gamete, two different kinds of gametes. So what I mean specifically is if that individual can produce eggs with either a Z or a W, we call it ZW sex determination. So if you can have the large and the small sex chromosome, an egg with a large or an egg with a small sex chromosome, we just call it ZW. And if it's the sperm producers that can make a sperm with a large or a small sex chromosome, we call that XY. That's the difference between ZW and XY. So in birds, they have ZW sex determination. It means it's the egg producers that can make an egg with a Z chromosome or a W chromosome. And then we move on down the phylogeny. And monotremes have a multi-X, multi-Y system. So platypus has 10 sex chromosomes. Echidnas have 9. Platypus has 5 X's and 5 Y's. Echidnas have 5 X's and 4 Y's. It's really fascinating. But the take home here is that the monotreme sex chromosomes are independently evolved from birds and the therian mammals. So in the common ancestor of all the therian mammals, we have a shared XY system. And then something different, right? I said there are these 3 major groups of mammals, and those 3 major groups have something unique each about their sex chromosome systems. The monotremes, totally independent sex chromosome system from the therians. The therians share this X conserved, Y conserved region. And then in the common ancestor of all eutherians, so us, dogs, pigs, whales, cats, mice, rats, we have an addition of a 50 million base pair region called the X and Y added region. That region is on a non-sex chromosome in marsupials and in monotremes. So thinking about our sex chromosomes, so this is 180 to 210 million years ago. We kind of hand wave with time estimates sometimes in evolutionary biology, but broadly, our X and Y chromosomes evolved from a pair of ordinary autosomes. They could recombine across their entire length. They were homologous, shared sequence content, just like chromosome pair 1, 2, 3, 4, 5. But sometime over the last 150 to 200 million years, those X and Y chromosomes evolved dramatically different gene content. So our X chromosome has about 155 million sites on it and 1,100 genes. And our Y chromosome has about 60 million sites on it and 27 unique genes. Some of those genes have many, many copies, but about 27 unique genes. Here's what I also like to point out. The Y chromosome, although it is small, it is not the smallest chromosome in our genome. It is the most gene poor, but it's not the smallest. So there's quite a lot of content on there, but coding content is pretty small. Doesn't mean there's not regulatory content there, or a bunch of repetitive content that we're not entirely sure what it does. And this is my favorite plot in the history of plots. It may seem simple, but I hope that you'll appreciate it as much as I do, at least groan about it for like a second as you're sitting there. Okay, so what I'm going to walk you through is the Y chromosome and the X chromosome and their sequence homology. This is a dot plot. This used to be very common when we were looking at sequence similarity between 2 chromosomes. And so you'll notice the X chromosome beautifully along the X axis and the Y chromosome here on the Y axis. Oh yeah, thank you for the shades. Right. Whoever put this together, I hope that they realized how much joy this would bring me and every person that I ever show it to from that Ross et al. Paper. It never gets old. And it's never old. I love it so much. So we have our X chromosome in sequence from the short arm to the long arm, and our Y chromosome is upside down here from the short arm to the long arm. And all these little dots on here are showing sequence similarity. But I'm going to walk you through that sequence similarity because it's a little difficult to see here. So the first part is that X added region. That's that 50 million base pair region I said was added in the common ancestor of all eutherian mammals. And what we can see are all of these little segments that align with high sequence similarity, we're getting these little dot plots showing sequence similarity between them. In particular, we have the pseudo-autosomal region, the pseudo-autosomal region 1, and it's not an autosome, but it does exist in 2 copies, one on the X, one on the Y, with 100% identity. So it's exactly identical between the X and the Y chromosome. So individuals with 2 X chromosomes have 2 copies of this, and individuals with an X and a Y have 2 exact copies of this. It's really difficult to see here and probably not even visible, but there's a second pseudo-autosomal region at the tip of the X and the Y chromosome. This is a human-specific duplication and translocation from the X to the Y. Additionally, there's another region of very high sequence similarity. It's called the X transposed region. So this is also a human-specific duplication from the X and translocation to the Y. And this region shares over 95% sequence identity between the X and the Y chromosome. So this is kind of anatomy of the X and the Y, and a reminder that they share an evolutionary origin and they share sequence similarity, but not 100% identity. So why does evolution matter? Well, evolution matters because of that shared sequence similarity, that shared homology. We anticipate misalignment of reads between the X and the Y for those sequences because our algorithms are designed to look for sequence similarity. That's great. They're doing exactly what they should be doing, but the X and Y are like, P.S. we actually share some sequence similarity and we're going to muck with your algorithms. And so, oh yeah, there's a comment about the platypus is so fascinating, and it is so cool. And if I weren't here to tell you something about X and Y chromosomes and the alignment algorithms we've been working on, I would gladly spend hours talking with any of you about the platypus. Sidebar, yes, I do have a platypus onesie. Yes, I have worn it to my child's school. Great. So here's a picture of our human chromosomes. And what I want to draw attention to is that we typically have 1 copy from genetic mom and 1 from genetic dad. And then individuals typically either have 2 X chromosomes or an X and a Y, right? I say typical because we have these other not uncommon and not abnormal circumstances where individuals will have 1 X or 2 X's and a Y or other configurations of the sex chromosomes. But typically individuals will have XX or XY. But here's our karyotype, our stained image of the chromosomes. And when we're doing genome alignment to a reference genome, we just use 1 representative of each chromosome. There are some software available to account for some of the haplotype differences or single nucleotide differences between those or that exist in a population, but largely we're still using 1 copy of each of the autosomes, and we include the entire X and the Y in our standard reference genome when we're doing our analysis. What's that mean? Oh gosh, okay, well that means that if you have an individual who is karyotypic, has an X and a Y chromosome, let's say chromosome 8, we take chromosome 8, chop it into short reads, and then we're going to align it to the reference genome. And when we align it to our standard reference genome, we take all the reads from chromosome 8 from genetic mom and chromosome 8 from genetic dad, and we align them to chromosome 8. We get all those reads, chopping there. When we take this individual's XY chromosomes, we chop them into short reads and sequence them, and then we align them to the standard reference genome, we anticipate that most of the reads are going to align to the X that are from the X, and reads will align to the Y for Y-specific reads, and we'll get a little bit of mismapping, especially in the pseudo-autosomal regions, which are 100% identical between the 2. What about our 46XX individual? So again, chromosome 8, we'll take that chromosome, align it to our reference genome, we get standard alignment. When we align chromosome X to our standard reference genome, what we've observed, and I'm going to show you, is that some of those reads from the X chromosome do map to the Y chromosome. So if you take out a BAM file from some of the major sequencing projects, you'll see that you get reads on the Y chromosome for everybody, for XX and XY individuals. It's not high read depth on the Y chromosome for your XX individuals, but it is read depth. And that's bad news bears, because those reads don't belong there. This individual, as far as we know, from all of the genetic and the phenotypic analysis, it doesn't have a Y chromosome. And if we went through and actually looked at gene expression, we wouldn't see anything for the Y chromosome, but those reads are mismapping now, right? Just because of sequence similarity. Oh. So what we did, this was really spearheaded by Tim Webster. He's a faculty at University of Utah now. If you're in the area, you should look him up. We did a hackathon at HackSeek. This was in Vancouver, gosh, forever ago, a lifetime ago, published in 2019, where we developed a software called XY Align to infer the sex chromosome complement of the sample, so whether it has a Y chromosome or not, output quality, depth, and allele balance across those windows, and realign with the appropriate sex chromosome masks. So there's nothing mind-altering about this. You could do this in any of your own pipelines, just that we put this together because we wanted to draw attention to the fact that X and Y are unique and they do need to be treated a little differently. And it's not something you can fix downstream. You have to fix it at the alignment step. So in particular, what we recommend or advocate here, a best case scenario right now is that if you have evidence of a Y chromosome, you should hard mask the pseudoautosomal regions and force those reads to go somewhere else in the genome. And what we find is typically they go to the other copy of the pseudoautosomal region on the X chromosome. And if there's no evidence of a Y chromosome, so no, we don't, in this case, we don't care how many X chromosomes someone has. We care, do they have evidence of a Y chromosome or not? And if there's no evidence of a Y, then you hard mask out the entire Y chromosome and force those reads to go where they align best. Most of them will go to the X, but occasionally some of them will go to an autosome. But we do find most of them go to the X chromosome. You should hard mask it rather than just remove it, because if you remove it, then combining your files for joint genotyping later or for transcriptome analysis later will get messed up. So we want to hard mask it just to keep the Y chromosome there, but not allow reads to align. Okay, so, I just converted to PDF at the last minute, and this is what I get. So read depth distinguishes XX from XY in whole genome sequence. So our individuals, I'm going to show you XX samples in red and XY samples in blue. And on the plot, this is going to be an autosome. We chose chromosome 19, read depth relative to X. And on the Y-axis, read depth on chromosome 19 relative to chromosome Y. And so in these individuals, when we just take the standard alignment, what we see is that we get read depth on the Y chromosome. For XY individuals, we get about twice the coverage of 19 relative to X. So it's not exactly up at 2, but it's close there. So we get about double the coverage of 19, which should have 2 copies. Chromosome 19 should have 2 relative to the 1 X in an XY individual. And then we get their Y, it's about 1, it's about the same. But you'll notice this axis is really skewed, and that's because of that small number of reads mapping to the Y chromosome in the XX individuals. And so we see some of them have very few reads. So the fewer reads you have mapping to the Y, the higher that ratio is going to be. And then the more reads you have mapping to the Y, the lower that ratio is going to be. So for this subset of samples, we see in whole genome data, we do get reads aligning to the Y, just to show you that it is happening. We repeated this for whole exome data, and we've done it, and I'll show you later, for transcriptome data, and we see the same thing, always that reads are aligning to the Y chromosome in an XX individual. This would suggest that there might be Y-linked reads in XX samples, but we don't think they're actually Y-linked reads. We just think they're mismapping between them. Additionally, I want to show you just one thing to think about with read balance at variable sites. So here we're going to say our reference is an A, and we have 2 reads mapping to A and 2 reads mapping to that site that have a T or a variation, a single nucleotide variation or a single nucleotide polymorphism. For an XX individual, we should expect about half of the DNA reads to come from one site and half to come from another, unless they're all non-referenced. So if they're monomorphic, let's say they're all T, then we expect all of the reads to look like 1 or non-reference. And so one of the things we can look at in our data on the autosomes is to see if we get about 50-50 read balance or something skewed. And on the X, do we get 50-50 in an XX individual, or do we get something different in an XY individual? So I'm going to walk you through a couple of the plots here with one main purpose, which is to tell you that we just don't trust the Y chromosome. That sneaky chromosome, there's so much we need to study about it, and it just doesn't fit everything. So most of this software, what we're doing is getting better calling on the X, and we are, full disclosure, not doing a heck of a lot better on the Y chromosome. We still need to actively work on this. So this is a read balance, and I'm not sure if it's coming through very well, but about 0.5 is right here. So it's saying for the autosomes, yeah, we're having a peak at variable sites of read balance of 0.5. And then you have some monomorphic sites here that are monomorphic non-reference. And this is true for XY and XX individuals. Great. Autosomes look like they have 1 copy from genetic mom, 1 from genetic dad. The X chromosome in our XX individuals, we again have that peak here around 0.5. And then we also have that non-reference monomorphic sites. And then our X chromosome in genetic males, for all those sites that we're calling, it should be about 1 because there's only a single X chromosome in this sample. So we don't see that peak. Y chromosome in a perfect world would look very much like this X chromosome plot for the XY individual would be shifted over. I already told you it doesn't. It's a mess in the most loving way. It is a mess in the most loving way. There's copy number variable sites on the Y chromosome. There's highly ampliconic regions that share sequence similarity of 97, 99% across in these large ampliconic regions across the Y chromosome. We are going to need to take a lot more focus before we can find a really easy way to fix our variant calling on the Y chromosome in a way that we can trust. Well, just letting you know that that's not this paper, and that's probably not for a few years down the pipeline. So just to show you how this changes, so I'll walk through the map quality. When we're aligning reads to an X chromosome from a standard reference genome, and then when we realign to a sex chromosome complement. And so it's maybe not super easy to see that we have pretty poor quality in the pseudoautosomal regions because those reads align identically to 2 different places on the X and the Y. And then this is that X transposed region where we're not doing really great. So let me just show you the difference between them because that makes it a little easier to see. So this is the difference, the map quality difference between our standard alignment and a sex chromosome complement specific alignment. And we can see that we just, massively improve those identical regions at the tips. And we do a pretty good job of improving read mapping quality in the X transposed region. And then there's, a little smattering of like, hey, we're doing good, yay, across the X chromosome. So with that, I also, for a single individual, we recalled SNPs. So we went through and we called variation with the standard approach and with the sex chromosome complement approach. And for that individual, we called just a massive number of SNPs in the pseudoautosomal regions. These we think are going to be really important, especially for samples that have lost an X or lost a Y, right? This applies to everybody that has X and Y chromosomes. Pretty much the whole population has an X. And then also, how many more SNPs do we call using a sex chromosome complement approach in the X transposed, X added, and X conserved region for a total of almost 10,000 more SNPs called in a single individual. We haven't done this yet on a very large scale to see what the effects are on variant calling across multiple individuals, but I anticipate that we will call much more variation across a population data set. And this is important if we're thinking about demography, if we're thinking about X-linked SNPs that might be important for autoimmune disease, which they've been identified in, for sex differences in cancer. Right. And then the one other thing I'll get through while we're here, and then we'll have plenty of time for questions, is thinking about how this misalignment affects gene expression estimates. So I'm kind of taking a throwback to microarray days. So during my PhD, we're looking at sex differences in gene expression using microarrays. And one of the things we had to do is go through all the spots on the Illumina array because some of the probes on the Illumina array for the X were identical to the probes that they were using for the Y. And that's because they just chopped up the X, chopped up the Y when they were looking at sequences, chose those sequences for X and Y, and didn't map to see that were they some of those identical regions between X and Y. And so we had to go through probe by probe and say, are these probes unique or not to the sex chromosomes? Well, the same thing applies to RNA-seq. So this is data that was led by a PhD student who just graduated. She's working at Mayo Clinic now, Kimberly Olney, and 3 undergraduates in the lab who've all gone on to grad school or med school. And what we did was look at the effects of your reference genome on transcriptome alignment and show that using a sex chromosome complement can increase your ability to detect sex differences in gene expression. So this is, again, I keep showing the X chromosome here along the X axis. And what I'm showing you here is just the increase in the reads or the fold change in transcriptome data. Previously, we were doing DNA, now it's transcriptome data. And we find that for transcriptome data, we're getting a lot of the reads, again, clustered in the pseudoautosomal regions at each of the tips and in the X transposed region. A little bit more aligning elsewhere on the X chromosome, but largely for RNA-seq, we're seeing it's really affecting individual genes and these 3 regions. In particular, here's just showing a zoom in of the pseudoautosomal region that we have in Windows, right? That we're missing transcripts in the pseudoautosomal region using a standard approach. This might not seem like there's a lot of differences between, this is our female to male, looking at the gene expression differences between females and males here, using a default or standard approach versus a sex chromosome complement approach. The biggest thing you'll notice is these blue Y-linked genes all change to 0 expression in genetic females. And that's because we're not allowing anything to align to the Y chromosome anymore. And if we look at the Venn diagram here, we see that we call, not a ton more, but we call 4 more genes as being differentially expressed between XX and XY samples, 1 on the X chromosome, and 3 on the Y. It should be trivial to say that the Y chromosome is differentially expressed between XX and XY individuals. But the fact that we don't call those 3 genes as being differentially expressed using a standard approach is problematic, right? We know the XX individuals don't have a Y chromosome, and yet we're still not calling those 3 genes because of sequence similarity. We're not calling them as differentially expressed. Right, we have thousands of genes across the genome, tens of thousands of genes. I'm pointing out a problem with a very small corner of it, but I guess the point of doing genomics is we really want to know what's going on everywhere. And if we're not including X and Y, then we're going to be missing things. What's really important is for a set of genes called gametologs. These are genes that existed on the ancestral autosome, and they still have a copy on X and Y. So they still have a functional copy on X. And we usually call it, for example, DDX3X, and they still have a functional copy on Y, DDX3Y, for example. And what I want to show you is how doing a sex chromosome complement informed alignment can affect our estimates of differential expression on these specific genes. So this is in some placenta data Michael mentioned that we're really interested in. This is still in prep, but I couldn't have asked for, like, every possible scenario is here, every possible scenario. So here's the standard approach and the sex chromosome complement. And here we're going to start out with boring, right? This gene is differentially expressed between XY and XX individuals, XY in green, XX in orange here. It's differentially expressed between them in a standard and a sex chromosome complement. You'll say, yeah, Melissa, I see there's like some differences, and yeah, there's maybe not as many, but it's still significantly different with multiple correction testing, right? So big deal. Okay, yeah, okay, you're right for this one, but let's look at the next one. We go from seeing no sex difference in differential expression to seeing a female bias in expression, a significant female bias in expression. So we go from, we have one example where we don't see any difference between the 2 reference genomes, they're still differentially expressed. And we go another example where we go from no sex difference to being differentially expressed. Case number 3, again, boring. No difference in the default or standard reference genome alignment, and no difference when we account for sex chromosome complement. 4th case is we go from a gene showing differential expression between males and females, and genetic males and genetic females, to showing no difference between genetic males and females. That's pretty cool too, I think. And then the last scenario is where we see a change in the difference. So we go from seeing a male bias in gene expression to a female bias, a significant female bias in the differential expression. So these are genes that have variable levels of sequence homology. And so doing your sex chromosome complement informed alignment is going to have variable effects on them. But no doubt it will have effects on them. So take home is that sex chromosomes have a unique evolutionary history. This affects sequence alignment, mapping, everything downstream. So if you haven't heard it enough already in this conference, right, always know your data. Know your data. Don't trust data. Always look at it, visualize it, figure out what's going on with every one of the steps, and try not to make assumptions about what's going on with the data, but rather just really, really know your data. With that, I want to thank Tim and Kimberly, Sarah, Jocelyn, Valeria. This was funded by an NIH R35 grant from NIGMS and support from the School of Life Sciences and the Center for Evolution and Medicine. And so with that, thank you all so much. I see there's some Q&A, but oh my gosh, there's so much. So Michael, you let me know how I should start.
Sure. I can, so I can bring the questions on. So let me, I'm gonna start, here's one from Adine, and I can just read these out loud for you. So how do the difficulties in XY alignment impact counts for cancer or testes genes which have been implicated in many cancers?
Yeah, so CT, those cancer testes genes and cancer testes antigens are a subset of those genes on the X chromosome in particular that they're expressed in. So for people who aren't aware of them, they're expressed in highly in the testis and they're expressed highly in cancer also. And so we haven't gone in and pulled out the cancer testes genes in particular, but depending on their sequence similarity, if they have any homology with the Y chromosome, it will affect them. If there are duplicate genes that are similar just within a chromosome, that's not something we yet address here, but that's what we're aiming to do with the next iteration of the grant, is to look in particular on the Y chromosome and the X chromosome for these duplicated regions, which is going to be important for sex chromosomes and autosomes with highly duplicated regions. So that was gosh, I should have just said we don't know. That was my longer way of saying we haven't yet figured it out, but it's something that we're thinking about.
Great. And then there's a question about pseudo-aligners. So how can sex chromosome informed alignment, like you showed, be incorporated into pseudo-alignment approaches such as Salmon or Callisto for RNA-seq?
Yeah, so in our paper, the BMC sex differences paper, we also use Salmon. And although you pseudo-align outside of a reference genome, you still need to map it back to something at the end. And so we do see that if what you're mapping back to is a standard reference genome, you still get the same sort of bias. And so I didn't show you the Salmon. That's an excellent question, right? Pseudo-aligners don't solve the problem of mapping back to a reference genome when you have large chunks of sequences that are identical or very similar. So they are great. They're great. They're just not, they're not perfect.
Yeah, I guess I think they would, I'm thinking like RSEM even would, these methods like RSEM would just split. They should split it evenly if the sequence is identical.
Yep. Yeah, but we don't want to do that, right? Because then, because they're not, like for every autosome, they don't do it that way. So we just want to be cautious about how we're representing. And how we represent our sex chromosomes in the reference genome is, I think, affects how people think about them too.
Absolutely. So I've got some that I wrote in while we're going through, but people in the audience, now's your chance to ask about sex chromosomes and the platypus. So Here's so here's one that I thought of while you were talking. So beyond pipeline inertia, are there other non-trivial barriers to why people are not doing the way you've recommended?
I don't think so. So I think there's one step, which is if you want to do a quick alignment to infer whether the sample has a Y or not, that would be an extra time step. But other than that, so you could do a quick alignment, and then say, based on read depth threshold here, and we're working on that. We're working on a tool for that too, right? We're working on that, but we don't have it out yet. You could do it yourself. Just read depth threshold. We think there's a Y or not. And then you could go back and do it. So that might be the only practical hesitation, but you could just say, this sample was collected from an XX individual. We're going to assume there's no Y chromosome. You could make some assumptions and still you'd be doing better for 90% of the sample.
And that could be a quick alignment to the non-PAR of the Y.
Yep.
So that would be trivially fast.
It should be really fast, right? It should be very fast. And I think it's going to be really critical, especially for aging samples. So if you're studying aging or for cancer samples. So in multiple myeloma, for example, something like 90% of males with multiple myeloma have lost the Y chromosome in their sample. So we shouldn't be aligning the Y chromosome in those samples that have lost the Y chromosome. And in aging individuals, individuals can lose in their, it's been studied largely in blood, mainly because people don't want to give you a liver sample, when they're 20 and 25 and 30 and 40, but we can get blood samples across that time range. And we see that the Y chromosome can be lost in 20%, 30%, 40% of the blood cells. So we want to be careful when we're doing that. But otherwise, no, I don't, other than why are people still using HG19? I think that's the answer, right? Sorry, that wasn't snarky. And if you're still using HG19, I don't judge you for it. I understand inertia is really hard. It is. It's really hard to do something new. This is our pipeline. It works. It gets us what we need. Is it perfect? No. No pipelines are perfect. No models are perfect. But we can make them a little better.
And people can start reviewing and making it hurt if people do the wrong thing in a situation where it would really make a mistake. So there's another question from James Walters. For the few Y-linked genes that did not show differential expression between XX and XY, could that be due to low read count and low statistical power, not a mapping issue in that case?
Jamie, I think it's very nice to see you. You're an icon. Yes. Well, I would say it's still a mapping issue for the Y-linked genes that change from being not differentially expressed to differentially expressed. I think we don't have the statistical power in the standard reference genome. For others, right, others are just not expressed, right? We have 27 unique Y genes in the humans. Some of them are testis specific. They're never expressed in other tissues that we look at. So they're just not expressed there at all. But generally, if the read count's that low, we would just filter it out as saying it's not expressed. But yeah, I think that's a really good point. And again, it's affecting a very small number of coding genes in the genome. The variant calling, I think, is a bigger issue for DNA.
Here's a question from Andrew McDavid. How much does copy number variation or larger structural variation complicate the mask depending on XY approach?
I guess it's that large copy number variation and structural variation affects the entire genome. And there's people that are working on that. And I guess I would hope that they'll think about how to incorporate X and Y into that. But yes, as the reason I showed that allele balance plot on the Y is because there's so much copy number and structural variation on the Y chromosome that it mucks up everything. And we need to really focus on it and probably work really closely with people who focus on copy number and structural variation there. Yeah, that's an excellent point.
I have one more question for myself. Let's see. Oh, here comes a question from. Here's a question from Emily Schwinger. Do you foresee that we will move to iterative masking approaches for alignment in general, especially now with long read sequencing and greater appreciation of structural variation between individuals?
I think that I think that's an excellent question, Emily. I think we will when we can get access to high computing, like cloud computing resources for cheap across large scale data sets. I think right now there's still a bit of a, personally, this might, right, I don't know from anybody else, but what I think is that in general, the reason we run these pipelines once is we know they work and we get to the end. And the time it takes to compute the alignment, to do the variant calling, to do the joint genotyping is pretty heavy. But when every institution can have access to high performance computing cheaply, NIH, that was a real cough at first, but then it was a real one. When we can have access to those resources cheaply, then I think we can work that iteration into our pipelines and people can redo it. Absolutely. I don't know when that will be. But it will come eventually. Oh, he's fine.
Now they're coming a little bit faster. So here's another, here's a question from Hannah Ramey. How do you suggest we deal with, or do we need to deal with, incomplete dosage compensation, X inactivation for X-linked genes?
Oh, Hannah, that's great. We're looking at X inactivation in the placenta. And the reason we wanted to do this alignment is because we want to make sure we call as many variants as possible on the X. So when we infer X inactivation status from DNA and RNA-seq data, we sequence the exome, we align it to our reference genome, we sequence the transcriptome and align it. If we've missed variants on the X chromosome because we have lower coverage because we're throwing away reads, we won't call as many variants on the X. We won't have the power to detect allele-specific expression because we just haven't called them there. So I don't think it matters so much that they're inactive or not. I think the reads will still be there, but if we want to study them, we have to do a good job of calling them. There you go. And I think we'll just miss the reads in general and miss the genes, right? We're always limited to which genes are expressed and which genes have genetic variation in them. But otherwise, I don't think that, if I think about it, I'll take a screenshot and send it to you, but I don't think the inactivation will affect the alignment step.
Well, it looks like you've saturated all the questions of the audience. I just want to thank you again for this excellent keynote. I think it dovetailed with a lot of other work at the conference where, as bioinformaticians, we really appreciate getting into the nitty-gritty of the data, and here it makes a substantial difference on the biology and the findings. So that's a great story for the group to hear.
Oh, thank you.
Okay. Okay. Thanks, everyone, and I'll close out the session.
See you.